Good day, this is Professor Will, CBMD PhD. Today's Thursday, July 22nd, 2021. It is 10:52 and 4 seconds a.m. Eastern Standard Time. Today's research, chromosomes. So let's hop to it here. Chromosome separation under focus. Date July 21st, 2021. Source, Université de Geneva. And during cell division, chromosomes are duplicated and separated so that one copy of each chromosome is inherited by each of the two emerging daughter cells. Correct distribution of chromosomes requires high accuracy, and defects in this process can cause aberrant distribution of chromosomes and facilitate cancer development. By analyzing the structure of the protein responsible for chromosome separation, a team has shed light on the mechanisms controlling this essential player in cell division. Before dividing, the cell duplicates its DNA and goes from single chromosomes with one arm to double chromosomes with two identical arms linked together by a ring-shaped protein complex cohesion. The two arms are then separated by the action of a molecular scissor, separars, which cuts a subunit to the cohesion complex to open up the ring. Once the chromosomes are separated, the cell divides and gives birth to two identical daughter cells. The cleavage of cohesion by separars is highly regulated. It must occur only at a very specific time during the cell cycle. And to achieve this, several inhibitory proteins independently block the activity of separars until the chromosomes have to be separated. However, up until now, the molecular mechanisms by which inhibitors control separars actively have remained elusive. Researchers and scientists use cryogenic electron microscopy, cryo-EM. This technique allows them to observe biological samples at very high resolution while maintaining them in their natural state. And by using this method, they were able to determine several structures of human separars in complex with one of its inhibitors, revealing new regulatory mechanisms for the enzyme. It turns out that these inhibitors occupy sites that also recognize the cohesion substrate, blocking the cleavage activity of the molecular scissors. While one of the inhibitors, securin, binds directly to the molecular scissors to block its active site, another inhibitor, the CCC complex, acts through a more sophisticated mechanism. By binding to the periphery of separars, the CCC complex induces a conformational change in separars itself. As a result, loops in separars, usually flexible and disordered, are recognized into a fixed position, leading to an autoinhibition of the enzyme. And this research significantly contributes to the understanding of mechanisms that regulate separars activation and could help design novel anti-cancer therapies. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.